thanks so much for coming and joining us. And also welcome to everyone on Zoom. There are quite a few people, which is great. May I introduce to you our visitor for this week? So we have Dr. Gabriela Hernández Mora. She came all the way from Costa Rica to spend a week with us. And she's the head of the Veterinary Medical Microbiology Unit of the National Animal Health Service of Costa Rica. And she's also affiliated with the National University of Costa Rica. And she's going to talk to us about Brucella in whales and dolphins. And for all the fish people who are joining us today, thanks so much for being here. And you will learn that Brucella can affect fish as well. So you made a good decision being here. All right, so I will hand over to Gabby.
So thank you for coming. Thank you for this wonderful week. Thank you, Kat. Thank you, Dr. Carmichael, to having me here. We're going to learn today about Brucella as a cause of strandings in the Eastern Tropical Pacific. This is the main animals, strandings, that we have in our country. Okay. So first, where is Costa Rica? So we are here in the middle of Central America. And so we have Pacific and Caribbean Sea. Okay. We have beautiful beaches. You're welcome to come. We have over 1,000 kilometers in the Pacific coast and 200 kilometers in our Caribbean coast. So it's quite work there when there is a stranding. We have a great marine biodiversity. We work currently with 80,000 animals, but we are expecting to have over 300,000 species in our coast, in our oceans. So we have quite a few marine biodiversity there. We are considered as oceanic oasis. So we have, for example, the thermic dome, which is in front of our coast. And so we have great visitors, like blue whales, big whales, during the year because of that amount of food around our oceans. So we got around 35% of the species of cetaceans of the world. That includes, of course, humpback whale that come from the north, the southern part, to reproduce in front of our coast. So we got a lot of moms and baby around. In total, we got 30 species in the Pacific and 30 species in the Caribbean. So, as you know, causal strandings are big. We can speak about natural causes, anthropogenic causes, and of course, like any other animal that gets sick, it can be affected by viruses, parasites, bacteria, and toxins. So cetaceans are considered sentinels of the health of the oceans. And it's important to think if something is able to kill one animal of 2-3 tons, it can affect humans, okay? We got diseases. We got common ancestors. We got common diseases. So why not Brucellosis, which is a classical bacterial disease in cows, why not affect cetaceans? So what is Brucella? Brucella is a bacteria. It is include like a gram-negative, non-motile cocobacilli, that's the shape of the bacteria, and is facultative intracellular. That is important because that means the bacteria can live outside the cells and inside the cells. If we think about this cell like an immune cell, normally this cell will detect bacteria like E. coli or Shigella or other classical bacteria, and it will destroy it. But what happened with Brucella? Well, and I'm calling her because in Spanish has a gender. She hides, okay? So the bacteria hides, Brucella hides, and not only that, it lives in the alive immune cells. So that is a really success bacteria. So this is the classical pattern of infection in ruminants, in terrestrial animals. So the bacteria enters through the different mucus, the eye, the nose, the mouth, and it distributed with different cells, within different cells, like polymorphonuclears, macrophages, and dendritic cells to the different organs, including lungs, liver, spleen. But the bacteria loves mammary gland. So when one animal is infected, the bacteria lives there, and of course, that milk is contaminated with the bacteria. So that is the main source for infection of humans normally, but also the bacteria loves the cells from the placenta. So depending on the amount of infection of the cells, it will cause the abortion, okay? And that's the other main source of infection for humans. There is different species of Brucella, though really the most dangerous ones for humans is Brucella melitensis, which the natural host is sheep and goats. Brucella abortus is one of the main causes like in America that we work with, and it infects cows. Brucella suis infect pigs and different wildlife. Brucella canis infect dogs. And Brucella seti infect cetaceans, and Brucella pinnipedalis infect pinnipeds. Brucella ovis affect sheep, and Brucella neotome infect rats and other rodents, as well as microti. There is other species that is in process for acceptance. And we in Costa Rica described recently a new one that affect bats. We call it Brucella nosferati. So Brucella change its anatomy. And you can see here a classical bacteria with LPS, lipopolysaccharide, and other components of the membrane that are classical. But these bacteria change a lot of the components of the membrane. They don't have flagella. The lipopolysaccharide is pretty abnormal. And so that is the components that help the bacteria to hide for the immune system. So if you infect one mice with 1 million of salmonellas, 24 hours later, the animal will have diarrhea. You can see the animal is sick. You can see the hair. He is not feeling well. But if you infect the same amount of brucellas in one mice, 24 hours later, the animal is perfect. So that's what happened in humans too and other animals. The immune system do not recognize the infection, and it's perfectly normal. And so it hides. She hides really well. So that's what she does. For example, viruses. We know that viruses need cells. So viruses go cell by cell infecting and surviving. But Brucella infect only some cells of the tissue, only some parts of the organ. So it's not homogeneously distributed in the organ. What about the transmission? So Brucella abortus, of course, is present in abortions in trillions of bacteria. This is that nasty picture for the ones that are not in this bloody and nasty world with samples. But this is the placenta. This is a testicle of a duck. You can see here the parenchyma, the functional tissue that is completely destroyed. We call it liquefied. And you can see the inflammation here in the epididymis that is really strong, severe. Also in the milk, and this is a placenta actually from a Kogia which is actually, you can see here the hemorrhage and other infections. And this is a testicle of a dolphin. So no matter the species, we can see that it affects the reproductive organs, okay? So we can say the transmission is direct by ingestion, milking, inhalation, transplacental, and reproductive, okay? What cause in humans? So we take the milk or get infected, and we start to have fever, like some chilling, some inappetence, pain in the back, in the bones, in the muscle. And these symptoms are in cyclone. We call it that undular fever in humans. That means that comes and goes, 1 week well, 1 week more, like weaker or stronger. And that, after all this time, it will come like more, in cycle. So you can spend years with the infection. Once the human get infected, the bacteria goes to different organs. That includes glands, liver, spleen, heart, causing endocarditis, testicle inflammation, some pain in the articulations, in the muscle, and if not treated, if not diagnosed, the bacteria can enter the brain. And from the point of the infection and to all this happen can last for years. So you will remember the real source of the infection. Let's talk about the marine part. So the ancestors of Brucella live in the grass, in the soil. So when these guys decide to go into the ocean, they already have the Brucella. We know that because of the molecular clocks that we have performed in the bacteria, the common ancestors of both terrestrial and marine bacteria. So Brucella have been there always, but we didn't know, okay? The history of marine brucellosis started in 1994 when, where in US, in some tour shops captive, they aborted, and in the same time of period, there were some researchers looking for some affections in strandings in UK. So they described something similar to Brucella, but by then, we didn't know that exists. So we call it at the beginning Brucella maris because we thought it was only one species. But then we at least know there is two recognized species, Brucella seti and Brucella pinipialis. Is more complex than that. We're gonna talk about in a bit. So in 2013, we performed this map, collecting all the information that was published by then. And so we have at least 35 different cetacean species with infections, with isolation, with pathologies related around the world. That means it's been there, like around in different continents. This is more recently, we performed this map, and the classification is a little complex now. So you can see all these numbers is how we classify the marine brucella. And that is because somehow in terrestrial animals, we have like look for the better cow, the pretty cow, the most pretty odor in the cow, the milking, but in wildlife, it's been free for millions of years. So the taxonomy is a little complex. So the ST, which is the black squares here, is how we classify sequence type. So in, for example, in Costa Rica, we describe 26, which is the most common, and 27. 27 is important because we're going to talk about that in a few slides. The cases of humans infected with marine brucella have been this type, the 27 type, okay? So all this is happening in all different species, okay? How these affect cetaceans. So we already know in cows, in terrestrial, in ruminants, it affects mainly the reproductive organs. But in humans, we have more pathologies related. These guys have both patterns, like natural host and accidental host. So we can see how these animals strand, at least in Costa Rica. These animals present, most of them, meningoencephalomyelitis, secondary hydrocephalus, and this happened because the bacteria is there making inflammation. So the normal flow of the CSF get stuck in some point of the inflammation, and the CSF start to accumulate here in the ventricle. So that is completely abnormal. We can see some hemorrhage here and all the affection in the brain, within the brain. These animals also strand with endocarditis, like humans. Also placentitis, those that are pregnant, and also osteoarthritis. The atlanto-occipital joint is one of the main with human, compared with human, because this articulation probably is more related to the brain, more close. So this articulation is important to study. So we know in terrestrial, there is some like possibility of infecting between species. So for example, Brucella melitensis can be transmitted to humans, Brucella abortus too, Brucella canis is weaker, but it can happen too, and Brucella suis. Brucella suis. Brucella melitensis also can infect dogs and also can infect other animals. Brucellan fish, I know you're happy. So fish are susceptible to brucellosis. I summarize this study of serological and bacteriological response in Nile catfish after experimental subcutaneous infection with Brucella melitensis developed antibodies 7 days post-inoculation with maintenance of positive titers for 5 weeks. That means infection. So the bacterium was subsequently isolated from various organs. So the bacteria can infect different organs from fish. Also, Brucella melitensis have been isolated from internal organs and skin swabs of seropositive Nile catfish naturally infected in the delta region of Nile in Egypt, Nile River in Egypt. So it is not known what role the fish may have, but in transmission or are reservoirs of infection in aquatic environments. However, marine brucella have been identified in cases of brucellosis in humans who frequently consume raw seafood. Okay, we're going to talk about them, these cases. So there's 4 known cases. So we said, not only 4. That's not dangerous, but there's a lot of investigation that need to be performed. The first infection in humans described with marine Brucella was in a laboratory worker. Of course, we are exposed to pure culture. And there was 3 other cases that have been reported. The epidemiologists do not recognize any of them being significant exposure to marine mammals, but all of them eat raw seafood, like good Peruvians in ceviche, and one fisherman here that used to eat fresh caught raw fish, okay? What they present, the guy from the lab presented headache, fatigue, and severe sinusitis. The ones from Peru had periorbital pain, that means pain around the eye, headache, seizures, and the other one had presented headache, nauseous, vomit, and progressive deterioration of the sight. So this is like more brain related. The one that affect in New Zealand have fever and lumbar pain, so more osteoarthritis and osteoarticular problems. So this is important because probably there's a lot of more humans infected around the world. But what happened? Well, the contact, like us, occurs in remote areas where the common symptoms of the disease may be confused with chikungunya, especially in our countries like dengue, chikungunya, all that. In these places, there's limited medical services. There's not a standardized or validated test for humans infected with this marine brucella. There is different report systems for human cases around the world. That its main difficulties to compare globally. There's not a specialized laboratory with marine brucella. And like you guys here in US, they eradicate or control the disease so that microbiologists is not used to see and work with these bacteria. So the expertise in animal laboratories is not routinely performed. Okay, and this is a new slide in my presentation after this week. We don't know, the scavenging in some parts like here is important. So we don't know if this marine brucella can affect the coyotes, the foxes. What about the domestic animals? We don't know. We don't know the sharks, and we know they are infected with ST27, so we don't know actually that infection that could happen in humans being exposed to this. So this is important too, to study. Brucella in race. Also, you're happy. So there is a new Brucella, a typical Brucella that was recently in 2000, well, not recent, 17, was described in race. So it's really worth to try different species. We are really like thinking in mammals all the time, but it's good to open our minds, and the cycle in marine environments is have not been described completely. So be aware of that. What we know about marine brucella in other animals, well, cattle experimentally infected with brucella, isolated from harbor purpose, demonstrate seroconversion and abortion under experimental conditions. So marine brucella can affect cattle. Brucella septi was less virulent than ruminants brucella in cows and rodents, other study. Seals are important part of the diet of polar bears, the later showing brucella seroprevalence between 5 and 10% in the Arctic. And also in Alaskan otter, there is a seroprevalence of brucella between 3 to 8% and 6% in the southern sea otter. So there's another mammals that can be infected, potentially with problems. And sorry about this, but I need that you remember this. So this is brucella in humans, can affect the brain. It's not a game, can be there, okay? Of course, this is Brucella melitensis. It's not that common. It happened in 5% of the human infected with brucella in the brain. So this is a brain of pretty more dolphin. So this is a brain of our animals. You can see here, even the impressions of that skull in the brain. That means it's swallowed. Once you take the CSF out, it's like a soccer game, like a soccer ball. It's deflated, okay? So these animals present neurological disease, and here in the US, you have the description of hearing loss in some of these animals. So imagine these animals with problems of hearing, what could happen. So this is a TAC CT scan. So this is a normal a brain in one animal negative for Brucella, and this is actually a brain of an animal with accumulation of CSF. So you can see, imagine all these making pressure of this brain against the wall, against the skull. If you have like a liver inflammation, you have like stomach to get extended. But the brain inside the skull, there's no way that you can extend that skull. So it's pressure, direct pressure. I pretty much try to think like the pain that these animals present is really severe. So this is the endocarditis that we describe. Is not common, but it happened. So for those that are not vets, the brown parts are bacteria within the cells of the tissue. So this is a lot of bacteria, Brucella, marine Brucella, in the tissue of this animal, in the heart. This is a picture from here. So also in arthritis, this is a joint nasty material. And this is one of our animals in Costa Rica. You can see here the hole in the atlanto-occipital joint. We were like, why? Why there is a hole there? So we take a swab, direct swab in the tissue, and there was pure culture of Brucella. So Brucella can make holes in the bones too. This is one of the few cases that have been described also. This is a narwhal. And so it causes steatitis, what means inflammation of the fatty tissue, also in this animal. This is rare, but it happened. This is a testicle from here, from animals from here, from US, and this is a uterus. Also, all these purple spots are inflammatory cells. So probably the inflammation causes fertility effects. This is a picture from our placentas. You can see all these white spots that are infection spots of Brucella. In this case, ST26, the classification. So the bacteria also cause placentitis and fetal loss and abortion. And this is the placenta from our koja. It's ST27. Remember, this is important for human infection. And all these creamy secretions are abnormal, and all this hemorrhage is abnormal too, so it causes abortion. And this is pretty. Remember, the brown is the bacteria, so this is the placenta of the koja, so it's highly infected. And again, remember, it's intracellular bacteria, so it's in the inflammatory cells where the bacteria is. So this is a picture from here, from US. I would say like probably is here from the Gulf of Mexico, this picture, because it was for like by that time that Katie gave us this picture. And so it caused abortions, okay? What we know about transmission. So inland, in farms, the bacteria survive direct contact with UV 4 to 5 hours. In places where it's humidity and organic material, it can last for months, 2 months. In poop, 3 months. In meat, is short because of the pH of the meat. We don't know in water, in seawater, how these bacteria survive, how long it survives. We don't know. We don't know. We know that the bacteria is present in millions, trillions in fetal liquids and tissue, aborted tissue. So this is like the most common infection for humans again. So it has, of course, horizontal transmission, vaginal fluids, semen can be transmitted, the bacteria. We isolate the bacteria from those fluids during pregnancy also, and it's important to think about this material during parturition, because other females, other groups, other members of the group can be in contact with that fluid. Remember that the bacteria enters through the eye, the mouth, and through the nose, in that case blowhole, but these fluids is important during parturition, and other helpers of the delivery could be exposed. Could be milk. We isolate the bacteria from milk of striped dolphins, so milk is important too. And this is really personally new, but this guy, this pathologist here from US, described really well the infection of paraphilaroides, is a lungworm, and Brucella pinnipedalis. Again, he performed the immunohistochemistry, and again, all these brown parts are positive for bacteria. So that nematodes, which is completely different from what we see in terrestrial animals, can have the bacteria in the gonads, and not only that, it can be transmitted to the larvae. Okay, and I was really pretty skeptical, like like one lungworm that come out from a dolphin and swim and go inside another dolphin is really weird. This infection. So we perform our like experiment. This is one of our lungs with lungworms in Costa Rica. And we like clean that and mash, and we got pure culture in our version with Allocercus and Brucellosis. So it's there. Like the role of these parasites is not known, but the Brucella is there and in big amounts. Not all the animals that have these parasite, the parasite are with Brucella. So we have like different, we got animals within like neurobrucellosis having parasites or not, and the parasite may have it or not. So we have different um, possibilities there. How to make the Brucella diagnosis, and this is really important. We can perform serology. We need blood and serum, and also CSF. We can make culture from tissues and fluids. We can use CSF, brain, milk, semen, abscesses, placenta. We can make histopathology. We can make immunohistochemistry using tissues, brain, uterus, testicles, joint capsule. We can make PCR. We're going to talk about that in a minute. We can use brain, uterus, testicles. We can use even CT scan to see if they have abnormal, for example, brain. Culture requires level 2, not level 3, as often assumed, infrastructure. And recently, Brucella is not included in USDA selected agent anymore. So, and Brucella said it never had. So it's just a matter of work and talk about that. So let's start with this new concept. So bacteria culture is gold standard, okay? And versus PCR. So what happens with virus? Let's think about this like an organ and all this as a cell. So in each cell, there is 1,000 to million copies per cell of the virus. So viruses infect organs more homogeneously. But what happens with Brucella? And we know the bacteria likes to hide. So we got here a couple of cells infected with the bacteria. We're talking about 2-3 bacteria per cell. There's literature about that. And when there is bacteremia, that means a lot of bacteria is like circulating in the body. We're talking 88 Brucella per milliliter of blood. So what happens if we send this part to make a PCR? It will come false negative because we don't send the whole thing and maybe not the cells that are infected with Brucella. Okay? So Brucella hides in some cells with an organ. And the excretion is intermittent. Okay? So that means in the odor, for example, not all the milk will be infected, not all days. Okay? So with culture, and why is culture gold standard? Because we work with a chunk, the complete chunk. We're talking about here centimeters of sample versus milliliters using for PCR. So if we send the whole chunk and maybe some cells are infected, we got positive culture, okay? And the limited detection in this media we use is cetapharial is 1 colony formation unit. And also the sensitivity is 1 colony formation unit per gram. So we know this, okay? All right, so what the World Organization of Animal Health says about the PCR. Even though PCR is described as a mean of detection and can you see? An identification for Brucella, a standardized protocol is not yet described by the WOA. We're talking here even for terrestrial animals, so even less information for marine animals. What are the problems of PCR in Brucella? We are working with few DNA of Brucella per cell versus about 30 billion of host DNA. The rupture of Brucella is not easy to get the DNA, and also it needs SDS, which interferes with PCR. So this detergent is important because the rupture is not easy. The DNA of contaminants, like our samples that are not that really pretty most of the time, interferes also. There is complete validation. The complete validation is deficient even for terrestrial animals. the lack of proper validation of selected protocols. It's there. There's a lack of a lot of, depending on the species, depending on the organ, depends on the amount of bacteria. And also another problem is the low positive and negative control sample used for these validations. So all this together makes the Organization Animal Health say that. How we work in Costa Rica? Okay, so we work with serum using CSF and tissue. Serum, if it's available, we perform Rose Bengal, E-ELISA, C-ELISA, and rapid tests. If there is no serum because of the time, we work with complete blood. If it's available, we perform these rapid tests. If there is not complete blood available, we work with aqueous humor using rapid tests, okay? And also the complete blood can be accessed from muscle, from liver, from different organs, okay? If there is CSF possible, and this is the best sample for culture, we perform CETA and fiber culture and blood culture. This is a new approach because, oh, I think this is dying. There you go. Because the commercial blood culture media have a lot of antibiotics. So some strains, marine strains, will die in the presence of that amount of antibiotics. These media have moderate amount of antibiotics that will give you some way to work with this few amount of bacteria present in the organs. If there's CSF possible, we also perform serology on that. We perform Rose Bengal, E-ELISA, C-ELISA, and these rapid tests. And like the last option, we perform direct PCR, okay? If there is not CSF available, we work with atlanto-occipital fluid. There you go, it's dying. So what about the tissue? Especially brain is our goal, not only cerebellum. We perform with 9 different parts of brain. We work with that. We work also with lungworms, gonad fluids, abscesses. Again, we culture in CETA and fibril and blood agar medium. And direct PCR, we are lately using, and we know, and is the experience here too, these PCR have really low CTs because of what I have explained. The few amount of bacteria is present in that amount of cells, infected cells. Also, we perform histopathology, immunohistochemistry, and most recently, hybridization in situ. What you guys perform here in the US, you work with direct PCR and histopathology. So you are losing a lot of information because you are limited to only these 2 techniques. We have the work in some tests that are rapid tests because we perform the serology before to start the necropsy. There you go. So we perform this. We perform Rose Bengal if there's serum, or we perform this rapid test. So you can see here, no matter if it's what we got here, we got complete blood, or we have humor aqueous. You have here CSF and more, complete blood from the muscle. So no matter the sample, it becomes positive. We know in 4 minutes before the necropsy if the animal is positive or not. This is important for healthy reasons. Okay, so we perform the CT scan that I told you, the CSF from the brain mainly, and the brain, the lung nematodes, we can perform the culture and some immunohistochemistry. So in these media, we almost got pure culture all the time, no matter if it's code 2 or 3 animals, okay? And we then perform the PCR direct from this colony to classify the bacteria. We have accredited all this in our laboratories. We collect the sample, so we got a reference area for the Eastern Tropical Pacific, and with this approach, we got 72% Brucella isolation success in stranded animals, positive stranded animals, okay? I won't tell you that, but this is a summary of the presentation, the pathologist presentations around the world. So no matter if it's central nervous system, reproductive tract, reticuloendothelial tract, musculoskeletal system, integument, respiratory tract, cardiovascular system, urinary tract, those are the pathologies related. Here are the examples of the tissue that to be collected. The ST that have been described of these tissues and the species affected. So this is a summary of worldwide sampling. So you better like see this. Okay. Serology. Okay, the serology have some problems. We need, at least in tropical waters, I don't know the heat here, but probably is a little less, 16 hours post-mortem in order to have some serum, okay? Some pretty serum. You can perform either, okay, so we collect intracardiacally the blood. So we have a lot of blood. We perform rose bengal, we perform the ELISAs, and sometimes we perform the immunofluorescence. It's a cheap test. You can use it on the field, and no matter if it's a human, cow, horse, dolphin, dolphin, um, you can describe if there is a current infected with Brucella or the animal have been previously infected. Okay, that's the answer that you're gonna get, and it's cheap. It's really, really cheap. This will be the best approach for study populations and sort of prevalence of disease, but I mean, it's probably impossible to know that, like live population to know that. Okay, the culture is the gold standard. That is the confirmation of the infection of the bacteria using a good media and easy bacteriological test. You can confirm, and then you can use the PCR. The media is really important because, as you know, the samples are not always fresh, and so the bacteriology people don't like that, send that nasty samples and try to recover something. But the media is really important. So no matter what, we work collecting. It's not even funny because the light's there still. Okay, so we collect the CSF no matter the serology that we have. So even from these really bloody animals like sample brain, we can have pretty CSF. We can see here uh, um, ice froze CSF. So we collect the whole chunk of CSF. And in the atlanto-occipital joint, that's another source where we can collect that sample. That is really important. So let's talk about that. So what if Brucella seti is present in the CSF, but not in the cerebellum, and you send cerebellum for PCR? And this is actually one of our animals. And in this animal, we had 100 cc's of CSF. Okay, so what happened if you send that amount, or I don't know, like 5 cc's of CSF to the laboratory? If they don't spin, if they don't, there, all right, I will move then. I'll stay. Um, so if you don't spin that sample, and remember, Brucella hides within the cells. If you don't spin that sample, you will have 100 cc's dilution. So you need to spin the cells, and you look for this amount of cells, and there you make that culture. Otherwise, you won't have culture. So you can see it's not easy, but you really have to look for it in order to be success. We perform this is like the screening for colonies. This is Brucella that tell you is terrestrial or marine Brucella. They won't tell you the species. So we perform another PCR, which asks for 16 genes at the same time. And so this is our cluster here in the Pacific from Costa Rica, compared with the other clusters of marine Brucella in the world. Those black are the terrestrial strains. So we need to know like more about the bacteria that is circulating here in the Gulf of Mexico, because you can know about the ST classification, but there's a lot of virulence adaptation that we need to describe, a lot of information of different genes that we need to understand in order to really see like how much it affected and how much it changed in this geographical area. Okay, we perform also whole genome sequence, and we classify like P1. So yeah, and how is the time? Is it good? What is the time? Yeah. Okay. So we start in 2004, like working with these guys, the striped dolphins. We got 94% of our stranding happen in the Pacific, only 6% in the Atlantic. And this is our like coast stranded spots. So it's happening all over the coast in Costa Rica. We have more information in the Pacific again. 36% of our animals are striped dolphin, followed by tursiops and humpback whales. And what we know about that, well, Brucella is present in Cepheus, in tursiops, and spotted, and blue whale that I cannot access to the brain, so I cannot culture from it, but it was positive to serology. Rhesus, roof-tooth, Fraser is the only species that was described in the Caribbean, Coryacyma, common dolphin, and striped dolphin. So we got all these different pathologies related mainly with the striped dolphin, okay? And also common dolphin have neurobrucellosis. And the Koja is interesting because we got the ST27 from this species, okay? Also toxoplasma is present. We isolate from striped dolphin, also toxoplasma in one animal that also have neurobrucellosis. So why we have the success of Brucella in Costa Rica? Because we perform serology before the complete necropsy is performed, the bacteriology of fresh collected samples, code 2 and 3, and the use of non-commercial selective medium with fewer antibiotic sida and pharals. There is no vaccines. This is one question. There's no vaccines. The only vaccine that exists is only for cattle, not even for buffaloes or other ruminants. So there's no vaccines that we can run behind a group of dolphins and vaccinating. No, that will happen. And yeah, I'm here. Of course, this is a summary of like our work. All these people in Costa Rica have help, and other from outside Costa Rica. This is my email if you want to ask me something, but this is like a summary of our work. Thank you.